Hi, everyone. Thank you for joining us for this Meet the Author interview. Today, we're interviewing Dr. Lucas Trambaiolli. He is an instructor in psychiatry in the lab of Dr. Suzanne Haber at McLean Hospital and Harvard Medical School. His research combines non-human primate neuroanatomy and human neuroimaging to identify brain circuits affected in mood and anxiety disorders, discover novel biomarkers, and develop new therapies using real-time neuroimaging. Dr. Trambaiolli is an MPP editorial intern and a member of the MPP Special Projects team. Today, we're chatting with Dr. Trambaiolli about his recent paper published in MPP entitled Translation of Monosynaptic Circuits Underlying Amygdala fMRI Neurofeedback Training. So just jumping right into the study, Dr. Trambaiolli, this study and also your research in general investigate neuroimaging data to find commonalities in both non-human primate and human brains. Why do you think that this translational approach of using animal to human data is important?
Well, first of all, thanks for the opportunity to be interviewed today. I feel very honored. Regarding this question, I do believe, and we here in the lab in general, we believe that animal models can be very informative to human neuroimaging, especially when we remember that the current methods that we have for human imaging, they are all indirect measurements of brain activity or brain structure. So diffusion MRIs, the orientation of water molecules, functional MRI is mainly based on oxygen consumption and blood flow. So we are not directly measuring like the brain per se. So we're measuring indirectly measuring the brain activity and the brain structure. So when we bring information from animal models, especially non-human primates, where we can in the microscope directly delineate the structure, the organization of connections, and from electrophysiology, where we can directly measure brain activity, we can use that to inform what we are seeing in the human brain using the indirect methods, optimize these methods, and improve our interpretation of results and consequently our understanding of disorders and treatments, tasks, and etc.
Jumping right into that topic, your study identifies key regions in the brain with which the amygdala communicates in order to modulate the salience network, first of all, but also the default mode network. So
Mhm.
functionally speaking, right, like those are a lot of big words, but functionally speaking, how do you think that this communication may be impaired in psychiatric disorders? And how could modulating these methods of communication be advantageous in therapeutic interventions?
That's a great question. So the salience network is a network, is an attentional network that helps us to identify salient stimuli in our environment and guide attention and behaviors. The default mode network is the mind-wandering network where our brain is usually like thinking about random things. Um, in psychiatric disorders, it's not uncommon to see patients showing abnormal patterns in these networks. And if we think about the populations that we were looking at the study, the major population that we had was depression. And it's not uncommon that these patients will have bias towards negative stimuli. They will have higher attention to negative stimuli, negative feedback, negative information in the environment. And this is usually related to abnormal activity in the salience network. The same way, these patients, they usually ruminate information related to their symptoms or their issues that they are dealing at that time. And this is usually associated with abnormal activity in the default mode network. And one of the interests in this study is that neurofeedback protocols targeting the amygdala have been showing that when you modulate the amygdala, you are also modulating these two networks. And what we were interested in looking at was because these two networks are very big, are very large. We wanted to see if the amygdala is connected with the voxels, with the sublocations of these networks that are being modulated after the neurofeedback training, during and after the neurofeedback training. And we were able to identify that there are monosynaptic connections linking the amygdala with these structures, which makes sense to imagine a causal model that if you stimulate the amygdala, you are also modulating these networks, and this could be a potential therapeutic approach in the future.
In your study, you talk a lot about neurofeedback. So why do you think that this study is important for delineating the specific mechanisms of neurofeedback?
That's also a great question. Neurofeedback, as many of the current treatments that we have available in psychiatry, is something that we see that it works, but we do not really understand how it works. This also happens with a lot of psychopharmacological interventions, and delineating the circuits as we started doing this study is important to understand pathways of action that we can later study, for example, with electrophysiology or dynamic causal models to understand how neurofeedback targeting one single ROI can have larger effects in the brain. And the goal of this study was exactly to try to understand these anatomical connections, these anatomical circuits, so later we can move on to understanding the dynamic of the circuits and providing not only a functional, but a whole anatomical functional delineation of the mechanism of this approach. And although we focus on amygdala neurofeedback in this study, this is an approach that can be used for any neurofeedback protocol targeting other regions or multiple regions at the same time. We can delineate the network involved in that task, in that approach, and then from there start to understand the neurophysiology of that intervention.
That is incredibly important. Thank you so much for explaining your study in terms that are really approachable and understandable. Kind of moving away from your study, as we mentioned in our introduction, you are currently one of the MPP editorial interns for this year. So for anyone who's thinking of applying, do you have any advice for anyone who's interested in the position?
Yes. Wow. First, I want to highlight how great the NPP internship has been for me. I think that being inside of a high impact journal, understanding how decisions are made, how priorities are defined, and how the workflow happens in these journals is great. Not only being at the author side, but being inside of the journal, it's amazing to see how everything works in this side of things. My suggestion would be that your cover letter needs to highlight your motivation very well. You need to make clear why you want to learn this, and not saying only that's like, oh, I want to learn how this works because I want to have more papers published, but what is your motivation, how this will impact your career. And one thing that all the interns have is a project. So if you can come up with an idea of a project that you are planning to develop during your internship, that would also be great because we can discuss it during the interview process, and you can start ahead. If you're selected, you can already have like the first steps in mind for your future internship.
Wonderful advice. Thank you so much, Dr. Trambaiolli, for taking the time to speak with us today. For anyone watching, if you haven't already, please check out the article that's linked in the video description below. As always, feel free to comment or reach out on Twitter if you have any questions. And we'll also include some details on how to apply for the MPP internship in the video description below as well. Thank you again, Dr. Trambaiolli. I hope you have a great day.
Thank you for the opportunity and have a wonderful day.